and he's going to tell us about their work with the cell phones and building microscopes, in this case, super resolution. Benedict, the floor is yours.
We're not online. No?
Oscar, do I need to?
Benedict, you can start.
Okay, so, well, thank you very much for the nice introduction. And so I'm just following up what was already presented earlier. So now, instead of going, well, deeper into the resolution bit, we try to decrease the price. And we do that with everything which is, well, probably in your hand or in your pocket, the cell phone. And at first, I would like to advertise a little bit the open aspect, which was already presented in many of the very nice talks here. And so just to compare, like the old style kind of research where you have an idea, do some experiments, theory, and publish it later, and finally publish that in public domain. Well, in the very end, people are getting to know what you're doing. So the new approach would be to have everything open. So what's the benefit there? So in every stage of your research, people get to know what you're doing. They can eventually improve. So there's like some kind of a feedback loop, and you can improve the results and you can learn a lot. And so at every stage, people can know what you're doing at the very moment. And that's very beneficial. And I think there's no reason to think of why there's somebody stealing your work. And why I'm emphasizing that, well, that's basically what we do and what we learned a lot during the last 2 years in 2 different projects. It's the UC2 and the CellStorm, which I'm going to present right now. And also it's fighting the whole reproducibility crisis, which is going on at the moment, where you basically cannot really reproduce optical experiments in this, in particular. And what does it mean? So I just took one random publication out with some optics, which I think is some 2-photon device microscope. And of course, well, if you think you just replicate that into the lab, well, that's not really possible because you don't really know the distances and the components used because it's not really put in there. And then, well, when I started doing my apprenticeship as an electrician 10 years ago, I was facing kind of the same problem. So I had the task to let a simple LED blink. So the task you do is you develop your circuits, you start designing a PCB, you finally manufacture that, assemble the stuff, and eventually also program something. And in the end, you really pray that it's working because you never really know whether there's eventually some error in between. And then in 2006, there was something called an Arduino. It's a small microcontroller which you connect to USB, for example. And you basically can get the LED blink within 2 to 3 minutes rather than 2 to 3 weeks. So, and we were thinking, since we are working in optics and microscopy in particular, how could we eventually port this phenomena to optics? And most of you eventually already know this scheme. It's called Fourier Optics. And it's very nice, and it's already inherently modular, because what you see here, you have a light source, you have some lenses, eventually imaging the light source into the sample plane. You then re-image the sample with some optics here, namely the objective lens and the tube lens onto the camera. But the cool thing is that the different components here, they are ordered such that the focal planes are intersecting with each other. And that's very cool because you can then directly modularize that. And we did that. Maybe a few of you already have seen that. We published it yesterday, so lucky coincidence. And once you have these building blocks, you can easily stack them together. For example, having an inverted microscope, which you can use for cell biology, where you put it into high safety labs, prepare your sample, slide it in, and once you're done, you either put away your sample, or once the whole microscope is eventually contaminated, you can just throw away the whole thing because it's plastics and very low cost. Of course, you can also integrate your cell phone camera instead of a Raspberry Pi, for example, where you just use your camera to image something, or you can build any other optical device, for example, a light sheet microscope. And so our idea was at every stage that people should be able to replicate that. So we felt reminded that there was something called the Google Era project, where they thought it would be very nice to have your cell phone modularized so that people can eventually, well, develop their own modules like a camera. And they supported them, the users, with something like a Modular Developer Kit. And we did the same. So if you, for example, have an idea of building a 2-photon microscope, then you can get stuff from our website. You combine it the way that you eventually end up with some modules. You combine the modules, and in the end, there will be a setup created. And well, how does that work? Well, so we have already kind of a library of different components. This also includes more complicated stuff like Z stages, but it's very important to mention that also your idea counts. So you can really just put whatever you have into this cube and stack them together. And I'll make a very quick scenario. Well, so I see microscopes as the bridge between the two different, or more than two different disciplines, which are here physics, engineering, and biology and chemistry. And so in our case, there was somebody having an idea to observe the differentiation from monocytes to macrophages. And this should be done over a timescale of some weeks. And so we thought, well, we now built this microscope, we put it together, we started doing the experiments in an incubator, and finally there was the result that you can observe differentiating macrophages. And that was already quite nice, because this is a highly interdisciplinary approach. And then we were thinking, well, how could we eventually push this thing a little bit more? And since this session is called super resolution, of course, we're tackling not only the price, but also the resolution. And so we were thinking, well, okay, so 3D printed plastics is eventually not really stable and not super reproducible enough. So we came up with the idea to injection mold it. And so it's a little bit like Lego now. So we it's not held by magnets anymore, but rather with this form fit mechanism. And you can now stack the different pieces with puzzle pieces, which you can concatenate. And what you see here is just one example of a more sophisticated setup, which incorporates two different methods, namely ISM and SIM. I'll just briefly mention the SIM capabilities, which you see here. It's this it's a small module you assemble with a DMD, and it just does what SIM does. It projects a structured illumination, namely a SIM, a sinusoidal pattern, into the sample plane. Why is that useful? Well, you can do the trick of, well, knowing the pattern of this structured illumination here. And then with the knowledge of this pattern, you can increase the resolution by, well, the maximum a factor of 2. And so what you can see here is the very first result of brightness or bright field resolution of actin filament labeled HeLa cells, and here the SIM reconstruction. And it's not published yet as a paper, but we already put everything into our GitHub so that people eventually can reproduce that. So that's basically the first half of the talk. And Jonas Ries already gave a very nice introduction into single molecule localization microscopy. And it's very nice to see that high throughput and low cost imaging devices are making its way. And already a while ago, we were thinking, well, why shouldn't we just replace this very, very expensive EMCCT camera with a cell phone camera? And the reason why that's possible in our case was that this camera, which is a Huawei, had a monochromatic camera chip. So that means you get almost 4 times more photons at a price of €20 instead of 20,000 euro for just the camera module. And so we're thinking, well, let's just reconstruct the results. It didn't work. The reason was that, of course, cameras are doing all kinds of image processing to make the pictures looking nice. And we came up with the idea of having a neural network-based reconstruction algorithm back then to go from the bright field image to the super resolution image with D-storm in this case. I don't want to go too much into detail, but it's a pixel-to-pixel GAN, generative adversarial network, which helped to get a resolution of 80 nanometer. The problem, of course, is that you still need a very bulky microscope to eventually get the resolution. And so then there was something very handy coming in. It's called nanoscopy on the chip, or STOM on the chip. is based on an idea from people from Norway, from Tromsø, around Balpreet. And the idea is that you have a chip, photonic chip, which has a waveguide. And on this waveguide, which has a very high refractive index, you let cells stick and you label them fluorescently, for example. You couple in light here. And what you then have is an evanescent field, which is exciting your fluorophores in a very, very low Z dimension. And that means that you have a very fine sectioning and very high intensity to eventually bleach your fluorophore or bring them to the dark state so that they start blinking. Still, of course, the problem is that you need very high cost components. And we came up with the idea of having everything small and low cost. This is the device, which you see now. This is the self-some device, which we also recently published. It's worth around €1,000, including the cell phone and all the software which runs on the cell phone. And the trick here, you see already the couple light into this little chip here, where the cells are sitting on top, is that you use a small lens from, for example, a cell phone or a Blu-ray player, which can be controlled in nanometer precision. So really everything what works here is worth like €1,000 and can be reproduced very easily. And so what can it do? Well, it can do D-storm, what you see here with some HeLa cells. That's always a very nice example. And you can go down to a resolution of roughly 100 nanometer. And so we were thinking, what else can we do? Well, we can do live cell imaging, not a very nice result, but at least some movement here. You can do also SOFI-like imaging, where you use the ability to fluctuate intensity with some surf-based algorithms from Ricardo Enrique's lab. And then we came up, well, it should be autark, so that everything runs on the cell phone. And therefore, we programmed a small app, which has a pre-trained neural network to get the resolution enhancement like SOFI or surf, but on the device with very low computational footprint. And so what you see here is like some results from membrane labeled E. coli, where you then, compared to the non-super resolution image, see the membrane in the end. So, and where else could it be useful? Well, so you can really get nice imaging results for large field of use using D-storm here. And also, since this whole Corona pandemic is still around, we thought we should definitely try out to image the Corona virus inside high safety labs, because the device is so cheap, you can replicate that easily. And therefore, we put one of these devices. You can just carry it around with your bike, and took it to the hospital and imaged some antibody labeled viruses, what you see here. So that's the comparison of one virus in bright field and in D-storm, and then we could go down to sub 100 nanometer resolution in a high safety lab. Well, I think that was already quite a lot, and I hope you still bear with me. I still hope in the end that you see too. And since I'm emphasizing the openness of everything, so I'm very open for questions. And if you have if you're curious, you can visit the two links here, and I can also share them later. Everything what you just saw is documented to replicate easily. So, and with this, I would like to conclude and thank, of course, the whole group from Professor Heinzmann here in Jena. And looking forward for questions.
Thanks a lot, Benedict. Looking at the forum, I don't think we have questions yet, but I do have one. So what kind of cell phones do you recommend for this now? Because some of those you mentioned are probably not easy to get these days.
Well, that's a good question. So for the study, we solely relied on the Huawei phones because they had this monochromatic camera. I'm aware of different phones which also have monochromatic cameras. But we also tend to use low-cost CMOS sensors instead. So because the smartphone is nice because you have all the connectivity and software in one place, but reproducibility, especially in terms of firmware updates and so, is not really guaranteed, unfortunately.
Excellent. So I think we're running out of time, right? Should we now move on to the Meet the Speaker session?
Yeah, I think we can move there, and we can carry on the discussion in the speaker session. Thank you everyone for attending and for the questions.